been really delightful being here and speaking with everybody. I'm extremely pleased to be a Sinauer lecturer. I worked with Andy Sinauer. He got me started in writing textbooks, and he is a revolutionary. He really came with a vision of writing biology textbooks, and this is just some of the biology textbooks that are in his catalog. And if you look at them, you'll see that these are some of the most important, neuron to brain evolution, life, animal behavior. Some of the most important books have come out historically. He was the first person to work with E.O. Wilson on a book. He really had an idea of publishing quality biology books, and not in New York City, not in Boston, but in Sunderland, Massachusetts. And so I can't say enough about him. He's also quite a gambler. He asked me to write my textbook when I was a postdoc. Okay? He was willing to take incredible risks. And the other person, of course, here who has taken incredible risks is Lynn Maroulis. Oh, I love this quote. "I don't consider my ideas controversial. I consider them right." And I generally view her work in, like Einstein's, in special relativity and general relativity. Kind of special symbiotic relativity is the endosymbiosis story, which has been just, one of the really new pillars of modern biology. And then kind of the general relativity, the general symbiosis, the universal symbiosis, the notion that organisms come together, that organisms form each other, that there is no real individual organism. What had been thought as being peripheral, lichens, is normative. That's what we all are. So biology has often been a study of individuals. There's anatomical individuality, physiological individuality, developmental individuality, immune individuality, genetic individuality, and last, evolutionary individuality. Anatomical individuality is a notion that our body has a single genome and it integrates our cells into a wholeness. This is the individual that we think about when we say, oh, I know him, I know her. We know them as individuals. Physiological individuality, that there's integrated organ systems, all the circulatory system, the gut system, the muscular system, all integrated towards a common end, that our physiology joins us together to a common individuality. Developmental individuality, that we are the products of the fertilized egg. This is something that Thomas Huxley would emphasize. What it is an individual starts at the zygote, and all the cell types that are put together, that derive from the zygote, that's what makes the individual. Immune individuality, Burnett's type of individuality. If I were to put my skin on you, you would reject it. I am a different individual than you are. The immune system exists, according to this notion, to protect the individual, that genetically produced individual, that single body, against harmful other individuals from the outside environment. And there's genetic individuality. The genome of each cell comes from the sperm-egg fusion, and for all intents and purposes, we have every cell in our body having the same genome. This is the individuality, by the way, which one sees in the anti-abortion movement, that you are who you are at fertilization. That is when you become an individual. An evolutionary individuality. The genome provides phenotype for natural selection. It's individual selection. Individuals are that which get selected, whether they be organisms or genes. So biology has dealt a lot with this notion of individuality. Well, one of the things that I want to say is that all these forms of individuality have been called into question, and I think really very much undermined, by current research on symbiosis. And the new type of individual, there's a new type of individual being formed, and that individual has sometimes been called the holobiont, which is the animal plus its persistent microbial communities. So when you think of a cow, you usually think of that mammal, kind of an archetypal mammal. But actually a cow is a dead organism unless it has its microbes in its gut which allow it to eat the grass that it so frequently does. A cow cannot eat grass without its microbial community. Here we see the coral, and the bleaching of coral is when the coral loses its algal symbionts, its zooxanthellae. When temperature or other pressures cause the zooxanthellae to leave the coral, it bleaches and it dies. It can't get carbon resources anymore from the algae. This organism here, those of you who've read Lynn Maroulis' work know Mastotermes darwinensis, which is a termite. And what is its individuality? Well, you could call the hive its individuality. And one termite, one female queen in the hive, the reproductive unit of the hive. But this is a worker. And this worker, digests trees, homes, other things that made of wood. Only it can't without its symbiont over here, the Mixotricha paradoxica. And Mixotricha, as has been called by Lynn Maroulis and Dorian Sagan, the beast with 5 genomes. And its cilia on the outside are not cilia, they're spirochetes. It has a rod-shaped bacillus. It has a round bacteria as well, which seems to act as a mitochondria. So where is the individuality? It's a composite individuality. The holobiont, the animal plus its persistent microbial communities. So from a holobiont perspective, we have to really see the world in a different way. Basically, we are all lichens. We are all products of networks of symbioses, and I will argue that animals are not individuals. Now, when we talk about symbiosis, we talk about 2 basic types. Mutualism, where 2 organisms, different species, interact biologically in a relationship where each of these 2 biological agents receives benefits, usually increased reproductive output. And that's represented here by this orchid. And the beauty of the orchid is made possible by fungi. A fungus spore has to enter the orchid to allow it to germinate. Without the fungus, the orchid doesn't have the carbon resources to germinate. So this is a very important symbiosis in the plant world. And then there's parasitism, which is a kind of symbiosis between 2 organisms of different species where one benefits at the expense of the other, hence the tapeworm here. Okay, I want to go through the types of individuality and talk about how modern studies of symbiosis has undercut them. And the first one is anatomic individuality. This is a section of the hive where I grew up in New York City. This is New York City, 1984, Fumio Kitaoka. It's probably one of the few Japanese prints he taught at Pratt Institute, which mentions Frankfurters, sausages, and knishes. But one of the things that's really nice about this, those of you who used to live in New York, this was the mutual benefit life building. Mutual benefit life. That's basically the theme of this picture in this talk. And the notion of anatomical individuality, that an individual is an organized collective of cells derived from the same source, the fertilized egg, well, it's not true. Humans are holobionts, and here we have one of them over here. We're not anatomical individuals. Over 90% of the cells in our body are prokaryotic. They have specific locations. These are kind of the analyses of the communities in our airways, the microbes on our skin, and the left hand differs from the right hand, in our intestines, in our mouth. And it's sometimes been called, the symbionts have been called the 11th organ system. Actually, there are several ecosystems in our body. About 160 species per person. And for the whole human species, there are about 1,100 major microbial species that are part of our anatomy. If you want to talk genotype, about 150 times more bacterial genes than mammalian genes. So we are not anatomical individuals. We are made up of many different types of species. What about genetic individuality, where all the cells of the body have the same nuclear genome, replicates of the genome we received at fertilization? Well, using the Holobiont perspective, we're not genetic individuals either. Symbionts are part of our epigenetic heritage. They are a second system of genes. If I were to give this picture to my students in developmental biology and say, what is it? I hope they would say, oh, this is from a germarium of an insect, and it has the nurse cells here, the supporting cells, and what's being transferred into this cell is going to be the oocyte, and they're transferring material into it. And this red thing could be ribosomes, it could be bicoid mRNA, it could be nanos mRNA, it could be mitochondria. They're giving things to this oocyte here. And that's exactly right. That's exactly what it is. This is the oocyte. These are the nurse cells of the germaria. But what the red dots are Wolbachia bacteria. The cells are giving the oocyte bacteria. The bacteria goes through the germline. You inherit the bacteria as if you would inherit mitochondria. So there's direct transmission of, in this case, Wolbachia in arthropods such as Drosophila. Well, we don't get our bacteria through our germline. We get it by infection. We get horizontally transmitted bacteria. As soon as the amnion breaks, we get bacteria coming in from the female reproductive tract. We get it from suckling. We get it from hugging. We get Bacteroides. We get bifidobacteria. We get all these bacteria immediately as soon as the water sac breaks. And in so doing, we go from one symbiotic environment, namely that of the mother, to another symbiotic environment, that of the bacterial world. And so we just go from one to the next. So these are the main ways of getting your bacteria. So we have genes not only from our parents, we have genes from our parents' symbionts. This is a second way of inheritance. Now, if you look at animals without sex, you really start seeing some interesting things. This is work by Moran concerning the pea aphid. And in pea aphid, these are asexual. These are parthenogenetic populations. A female gives birth to another female, gives birth to another female, and so forth. You don't have males in many of the lineages here. Female generation. So where is variation coming from? In part, it's coming from symbionts. So in this case, without the symbiont Rickettsia, a red aphid remains red. But if the strain gets infected with Rickettsia, the red aphid, genetically red aphid, will turn green as adults. The Rickettsia actually induces the synthesis of quinones in the aphids. So here we have variation, and it's genetic variation, but it's not produced by the genome of the aphid itself, it's produced by the aphid symbiont, Rickettsia. Well, same aphid. You can have allelic differences in another symbiont, Buchnera. And Buchnera is involved, Buchnera has a gene which, if functional, produces a chaperonin protein. It produces a heat shock protein. And there's a trade-off between fecundity at lower temperatures, heat tolerance at higher temperatures. So here, the dark pink is the full transcript. This is the unmutated transcript. The short one, the light pink, this is the mutant over here. And those, the mutant is basically a loss of function mutant. And those without the chaperonin protein seem to do better at most temperatures. They have more offspring at most temperatures. However, at high temperatures, which are found in the wild, they have very few offspring, whereas those with the chaperonin proteins have the normal amount of offspring. So here again, here is genetically encoded variation, which is important for the aphid. But is it from the aphid's genome? Actually not. It's from its symbionts genome. And the last aphid story I want to talk to you about is parasitoid wasp infection. So here is these nasties, these parasitoid wasps that Darwin hated, injecting its egg into a larva of the aphid, and this egg is going to hatch within the aphid. It is going to form a larva which will eat the aphid from the inside out and then digest its cuticle and be free as a wasp. Okay, nasty wasp. Okay, the aphid has protection against it. It has a particular bacteria, Haemotinella, and Haemotinella defensa can protect. So here's percent parasitized. Okay, without Haemotinella, it gets parasitized. With Haemotinella, very little parasitism occurs. The bacteria is able to prevent parasitism, only it can't. The bacteria need to have in it a lysogenic phage. It's the phage that protects it. Without the phage, you don't get the protection. So basically, you have an aphid. It can't give protection alone against parasitism. It has a bacteria. The bacteria can't provide protection, but if the bacteria has a phage in it, the phage will produce protection against the wasp infection. Okay, so hereditary variation through symbionts, going through the germline of the aphid. Is there any such thing in humans? Is there, anything in mammals that might say that there's, differences between populations of bacteria and that the bacteria can give us different phenotypes? Maybe. In humans, Japanese population contains a different strain of Bacteroides plebeius than Western populations. The Japanese strain contains 2 genes which they got by horizontal transmission from a Bacteroides strain that lives on red algae. The Bacteroides strain in Japan allows people to get more calories out of sushi than in the Western strains of Bacteroides plebeius. So even Bacteroides plebeius, which is a very common symbiont in humans, Bacteroides, as we'll see, is incredibly important in humans. There are strain differences. There are allelic differences between symbiont strains, which may possibly cause phenotypic differences. Of that, we really don't know. Okay, developmental individuality. That an individual is defined by coming from the same fertilized egg. Well, here's where I actually want to extend one of Lynn Maroulis' theories. And she and Dorian Sagan have a wonderful article in a journal that came out from a Boston science meeting where they talk about autopoietic organisms and the notion that what is remarkable is the tendency of autopoietic entities to interact with other autopoietic entities. Here we have a beautiful example of those. If you don't know about this, you should know about this organism. It's a solar-powered sea slug. It's a mollusk which ingests the algae ingests the chloroplast from algae when it eats as a larvae, and when it becomes an adult, the algae go to its ectoderm and basically are able to be used by the organism to make sugars. So here we have an organism, an animal organism, which is using chloroplasts to make sugars from sunlight. Solar-powered sea slug. Okay, but what I want to say is that we have not been autopoietic entities. Autopoietic means that we can form ourselves. And what I say is, nah, we can't even do that. From the Holobiont perspective, animals are not autopoietic entities. We use instructions from our environment, such as diet, and from other species, the symbionts. And perhaps you see the optical illusion here. Here's a baby, here's the eyes, here's the hands, the feet. Okay. And what I want to say is the environment helps construct the organism. That's what I want to talk to you about. Okay, back to insects, back to these parasitoid wasps, different species of parasitoid wasp. Here we have a situation where if you look at the DNA in the egg of one of these wasps, you'll find the nucleus, okay? Here's the nucleus stained with propydium iodide. And in the posterior part of the egg, you find all this other DNA. This happens to be Wolbachia bacteria. Close up, here's the nucleus, here are the bacteria. Now you could take this wasp and put them on a diet of rifampicin, basically a bactericidal drug. It'll destroy bacteria. It's an antibiotic. And you destroy the Wolbachia. You get rid of the bacteria. And when you get rid of the bacteria, you get rid of the fly's ovaries. The fly's ovaries need the bacteria. They can't develop properly without the bacteria. If they don't have the bacteria, they undergo apoptosis. Here's the tunnel staining. This is looking at cells which are undergoing apoptosis. This is without the symbionts. This is with the symbionts. There's hardly any apoptosis with the symbionts. Nice data here. Here's symbionts. Here's without symbionts. Without symbionts, basically the female reproductive tract is destroyed. Well, okay, insects are fine. What about mammals? What we find in mammals is even more incredible, I think. We believe, and I used to teach in developmental biology, the environment, has to be of a certain permissive nature, but, the instructions for development are pretty much in our genes and cytoplasm. Only they're not. This is the intestinal blood vessels shown in green in a germ-free mouse. So this mouse is bred sterilely, does not have bacteria in its gut, and it has a very rudimentary gut circulation. This is the villi here, and you should have an extensive circulation to get the nutrients out of the intestinal villi. If you add the bacteria back, you get the normal circulatory system of the villus. You get the intestinal vessels forming normally if you add back the microbes within 10 days or so. So it looks like the microbes are responsible for causing the vasculature, which is important for getting nutrients out of our intestine. Similarly with our immune system. This is looking at the gut-associated lymphoid tissue, the dome and the follicle of the gut-associated lymphoid tissue. This is with the gut microbes. This is what happens under normal conditions when the mouse is raised with the normal gut microbes that it gets from its environment, largely from its mother. You have the dome and the follicle. These are the activated lymphocytes of the gut-associated lymphoid tissue. This is what happens without the bacteria. You don't get gut-associated lymphoid tissue if you don't have microbes. The microbes are responsible for activating the morphogenesis of the gut-associated lymphoid tissue. They're also responsible for expanding the B cell and T cell repertoire. Okay, so what's happening here? This is actually the paper that drew me into this whole field. The mRNAs of the intestinal epithelial cell are altered by the bacteria. If you look at these, this is relative mRNA abundance set to 1.0 in a germ-free animal. So in a germ-free animal, this is the animal derived from the fertilization event, set at 1.0. Okay? These are the mRNAs for colipase, involved in lipid transport, angiogenin 4, which is involved in blood vessel formation, SPRR2A, which is involved in forming a matrix between the gut and the epithelial cells and the lumen. Now, if we look at colipase, here's 1.0. This is the fertilization event, what happens germ-free. Here's conventionally raised. This is when you have bacteria, 6-fold more. And if you add gut bacteria back into the germ-free mice, you can get it back to normal levels, and Bacteroides is pretty much responsible for this. Angiogenin 4, 10-fold. This is like a mutant. This is like a mutant that's only making 10% the amount of mRNA as your normal. because this is the conventionally raised mouse. This is the conventionally raised mouse, except without bacteria. This is the mouse without bacteria. Again, Bacteroides is pretty much important. SPRR2A, 50-fold more increase in mRNA. So this is making only 2% the normal amount of mRNA, germ-free mice. We have over 100 different bacterial species in the human gut. It looks like we have been outsourcing developmental phenomena. These bacteria are going to be with us. We expect them to be with us. They can do something for us. What they do? They are involved in colipase, angiogenin, SPRR2A production. Angiogenin is fascinating. Angiogenin, it's the protein that helps make the blood vessels. That's what angiogenin means. But angiogenin does something else. Angiogenin, which is made by Bacteroides, it's stimulated by Bacteroides, kills Listeria. It kills Listeria bacteria. It's bactericidal against Listeria. Listeria is one of the biggest competitors of Bacteroides. So we mammals become the contract killers against Listeria. We're helping Bacteroides kill its competitor. And in so doing, helping ourselves. We don't want Listeria either. So we have a situation here where the bacteria is inducing normal gene expression. It's what we expect. In zebrafish, it's causing the stem cells to proliferate normally. This is the normal amount. This is when you have conventionally raised zebrafish. If you raise zebrafish germ-free, what you'll see over here, much fewer stem cell division than conventionally. The zebrafish expects bacteria to be there in order to have normal stem cell proliferation. It seems to be activated through the Wnt pathway. Germ-free zebrafish have intestines that have very little brush border, paucity of enteroendocrine and goblet cells because they don't have many stem cells that are dividing. So this is normative. In other words, we are not autopoietic animals. We actually need the symbionts for our normal development. We expect the symbionts. This has real important ramifications in medicine and conservation biology. This is manzanella, a roundworm. It's a parasite. It's related to the elephantiasis parasite. Doesn't give anywhere near as bad a lymphatic condition, but it does give lethargy, tiredness. Manzanella has become immune to the anti-Helminth drugs. Anti-worm drugs will not kill Manzanella anymore. It's become resistant to them. So how do you kill Manzanella? You use doxycycline, good old antibiotic, antimicrobial antibiotic, because if you can kill the symbionts of Manzanella, which are involved in its molting, you get rid of Manzanella. Don't kill the organism, kill its symbionts. You kill the holobiont the same way. Here's a spotted salamander in the American Midwest. Here's its egg mass. And you can see that its egg mass it's green. The reason it's green is the symbiont of the egg mass is called Oophila amblystomasis, which is, it's the lover of amphibian eggs. The only source of food for this algae is the egg jelly of amphibians. Well, here's the egg jelly, here's the algae, everything's fine. The algae is actually producing oxygen for the inner cells here, which are oxygen starved otherwise. It's producing carbon. That was shown a few months ago. It's producing carbon resources as well for the embryos to survive. But this is in the American Midwest. Runoff of herbicides from the streams are going into the ponds. The agricultural herbicides are doing what agricultural herbicides are supposed to do. They're killing algae. They're killing plant cells. And in killing the plant cells, they kill the salamander embryos because the salamander embryos can't get enough oxygen or nourishment. Here we see, this is, some of you had mentioned Brian Hall's work before. There's some recent work from Brian's lab. Here you see the salamander embryo over here. You can see the eye. Here are the gills. All this green here is algae. But by the time it's a norula here, the algae is actually going into the blastopore lip, to the latest part of the blastopore lip. And it goes into all the cells, and it especially goes into the female reproductive cell layer. And so when the female eventually undergoes metamorphosis, gets fertilized, lays her eggs, what happens is the algae come out along with the egg. It's a nice system. So we have here, again, developmental symbiosis. Kind of the poster organism for developmental symbiosis is Euprymna, the squid, the bottle squid of the shallow waters of Hawaii. The squid has a light organ. And those of you who've gone scuba diving in Hawaii have seen this squid. It has a light organ, and the light organ throws light on the shallow floor of the seabed so that the shadow from the moon doesn't arouse predators. So that basically the shadow would cause predators to know that the squid is there. This light organ eliminates the shadow. Okay, only Euprymna does not have a light organ. It's not born with a light organ. The ventral surface of Euprymna actually collects Vibrio fischeri bacteria from the environment. There's not many Vibrio fischeri per milliliter in the environment, but it collects those that are there. They multiply. They actually interact with the, in a very interesting way, they interact with the ventral surface. They cause cell death in the surface. They cause remodeling of the juvenile cell surface, and they make a light organ. The bacteria then enter the light organ. They reach a critical density, and they start luminescing. They start producing light. The light can actually be controlled by kind of iris that the squid has. So basically, this light organ is an organ which is made by the interaction of bacteria and the squid. Just like our gut-associated lymphocyte tissue is made in combination between us and the bacteria. The mammalian capillary system in the villi being made by a combination of bacteria and the mammalian organism. What I'm going to show you next borders on science fiction. If this doesn't border on science fiction. Normal gut microbiota modulate normal brain development and behavior. There has been the microbiota gut-brain axis. Several papers have termed this the microbiota gut-brain axis. If you look at germ-free mice, and you look at specific mice with specific types of bacteria in them, you'll notice that the mice with bacteria have large amounts of this transcription factor, NGF1A, which is involved in neural plasticity. They also have higher amounts of brain-derived neurotrophic factor, which is a paracrine factor involved in neural plasticity. Basically, this is the symbionts acting in brain development. Germ-free mice had elevated dopamine levels. Germ-free mice had lowered amounts of NGF1A and BDNF gene expression. And the authors here, Heitz et al., say, during evolution, the colonization of gut microbiota has become integrated into the programming of brain development, affecting motor control and anxiety-like behavior. Well, here we have another situation. Again, PNAS, but this is the ingestion of lactobacillus strain regulates emotional behavior and central GABA receptor expression in the mouse via the vagus nerve. Okay, basically what you have here is that if you have a control mouse and you give it broth, you don't get much corticosterone. It doesn't have much anxiety. If you stress the mouse and give it just a broth diet, fine, yeah, it has anxiety. Control with this lactobacillus strain, yeah, still like the control. You didn't stress it. But if you stress the mouse after giving it this particular lactobacillus, it has less corticosterone levels. Not only that, you can eliminate this difference by cutting the vagus nerve.